Clinical researchers studying distinct pathologies, from type 1 diabetes to Parkinson's, have documented a recurring biological pattern. They converge on a shared precursor. Across these diverse autoimmune and neurological conditions, patient data reveals an identical severe downward trend, a profound accretion of a bacterial guild known as firmicutes. This specific dysbiosis signature shows Akkermansia mucinifila collapsing alongside the firmicutes, while pro-inflammatory taxa sharply rise. This signature represents a missing metabolic function. Across these diverse cohorts, the common denominator is a failure of specific microbial production. The missing function driving this signature is the colonic production of short-chain fatty acids, or SCFAs. We can trace exactly how SCFA deficiency physically dismantles the gut barrier to trigger systemic inflammation, and how it drives the epigenetic collapse of the immune system's tolerance mechanisms. Effectively treating these diverse diseases requires restoring this lost metabolic ecosystem rather than focusing on the eradication of individual pathogens. A healthy adult colonic lumen operates as a high-volume bioreactor, producing 300 to 600 millimoles of short-chain fatty acids every day. This diagram illustrates how complex dietary fibers enter the colon and are broken down into a specific chemical stoichiometry, a 60 to 20-20 ratio of acetate, propionate, and butyrate. These metabolites are generated by specialized functional bacterial guilds working in concert. The Faecalibacterium guild produces the butyrate, while the Bacteroidetes and Akkermansia guild produces the propionate. When these specific microbial populations are depleted in a dysbiotic state, production halts completely, resulting in a catastrophic drop in SCFA output. Without the correct dietary substrate fueling these precise microbial guilds, the colon loses its primary metabolic currency. This localized starvation initiates a systemic biological breakdown. To see the first stage of this breakdown, we examine the human colonocyte wall. For these epithelial cells, butyrate is the primary fuel source. This comparison shows colonocyte metabolism. The healthy cell on the left derives approximately 70% of its operational energy directly from butyrate through beta oxidation. The deficient cell on the right is energy-starved. This energy requirement translates directly to mechanical failure. Without butyrate, the cell cannot sustain MuC2 expression, causing the protective mucus layer to dissolve. The energy deficit also degrades the structural integrity of tight junctions, specifically the proteins claudin-1 and occludin. Without metabolic fuel, these physical seals between adjacent cells fail. Once these seals break apart, the paracellular space opens. This physical breach is the mechanism clinically defined as paracellular permeability, or leaky gut. The danger lies in what passes through those gaps. Lipopolysaccharides, or LPS, and other microbial products translocate directly from the lumen into the bloodstream. This constant influx of foreign microbial products into the blood drives systemic LPS endotoxemia. SCFA deficiency is the physical root of the low-grade inflammation seen in NAFLD, multiple sclerosis, and type 1 diabetes. Beyond physical barrier maintenance, SCFAs serve as metabolic fuel while also acting as powerful epigenetic modulators that direct systemic immune regulation. We can trace this modulation directly to the naive CD4+ T cell, an immune cell awaiting biological instruction. This graphic shows butyrate entering the T cell nucleus. It physically docks into histone deacetylase enzymes, blocking their function in a mechanism known as HDAC inhibition. Blocking this enzyme physically uncoils the T cell's DNA at the FOXP3 promoter region. Activating this promoter forces the naive cell to differentiate into a peripheral regulatory T cell, or Treg. These cells serve as the body's primary defense against autoimmunity. While butyrate works inside the nucleus, a secondary immune mechanism operates at the cell membrane via G-protein-coupled receptors. The receptors GPR43 and GPR109A bind propionate and acetate from the bloodstream. This binding action sends chemical signals that directly constrain the expansion of inflammatory Th17 cells. Losing colonic SCFAs creates a tripartite failure. It starves the physical gut barrier, turns off anti-inflammatory receptor signaling, and removes the epigenetic instruction required to build autoimmune suppressing Tregs. If SCFA deficiency explains the pathophysiology of these diseases, it raises a clinical paradox. Why do standard oral sodium butyrate pills consistently fail in human trials? This diagram of the digestive tract highlights the pharmacokinetic problem. Free oral SCFAs are absorbed entirely in the proximal small intestine. Because these supplements never reach the colon, they cannot exert the localized barrier healing or epigenetic effects where the tissue requires them. Single-strain probiotic therapies face a similar hurdle. Isolated bacteria cannot survive or colonize without their specific companion strains and the dietary substrates they require to ferment. You cannot treat a functional ecosystem collapse with a single pill or a single bacterial strain. Restoring this metabolic output requires comprehensive ecosystem engineering. This requires a multi-pronged ecological intervention specifically designed to bypass early absorption and rebuild the microbiome from the ground up. The first component relies on prebiotic substrates engineered for targeted colonic delivery. This visualization tracks HAMSAB, high amylose maize-resistant starch, as it bypasses the small intestine, releasing its payload only upon reaching the colon. Once in the colon, this substrate acts as targeted fuel. It selectively feeds the endogenously depleted SCFA-producing bacteria already present in the patient's gut. The second component introduces keystone next-generation probiotics. By seeding taxa like Akkermansia and Faecalibacterium prosnitziae, the protocol forcefully reestablishes the mucus layer and scales up butyrate production. Sustaining this newly planted ecosystem requires a dietary scaffold, such as a high-fiber Mediterranean diet, to maintain continuous substrate volume. Executed together, the cascade is exact. Targeted substrate reaches the colon, keystone species multiply, SCFA ratios are restored, tight junctions seal, and immune-tolerizing Tregs differentiate. Reversing the trans-disease dysbiosis signature requires matching precise metabolic substrates to missing microbial functions, reestablishing the specific metabolic pathways that maintain systemic immune tolerance.